Hello, my name is Athena Allred, and I worked in Dr. Bannock's physiology lab where they are looking at a new treatment for polycystic kidney disease, or PKD. PKD is an inheritable and incurable form of kidney dysfunction in which fluid-filled cysts systematically overtake healthy tissue, leading to eventual kidney failure. I was tasked with answering the question of whether or not PKD is affected by the presence of nerves. To do this, I examined two sets of kidneys, one untreated with the nerves intact, and the other treated with the nerves cut. Using image analysis software, I measured the total area of the kidney and the total non-cystic area of the kidney, and plugged those numbers into an equation that told me what percent of the kidney was cystic tissue. I also examined microscopic images to determine the amount of glomerular fibrosis in the kidney tissue, which is an indication of inflammation that is common in PKD patients. We hypothesized that the treated kidney would have lower levels of glomerular fibrosis and cystic progression when compared with the untreated kidney. However, our results did not support our hypothesis because there was little to no significance between the treated and untreated kidneys. While this was a negative study, these results may support the further hypothesis that renal denervation helps to slow the progression of PKD through a systemic rather than local effect. Further research is still needed into how renal denervation can help to slow the progression of the disease. Thank you for listening.
Hi, my name is Amel, and this summer in the Gutriff Lab, I explored the importance of biomaterial property considerations in the development of implantable and wearable devices for sensing and stimulation needs. And one challenge these devices face is everyday muscle and joint movement, which can stretch the biomaterial and eventually plastically deform it, which can lead to breakage failures in the device. And one way we can begin to address these concerns is by analyzing stress-strain curves. My project focuses on evaluating material properties such as deformation behavior, tensile strength, which is the maximum stress a material can endure before fracture, and elastic modulus, which defines how stiff or soft the material is. I specifically tested thermoplastic polyurethane, or TPU, and eel and their doped counterparts to define their relative strengths and elongation capacities. To do this, I utilized the Astera stretching stage to induce strain onto our samples with the intent of cleanly breaking them. And additionally, I cycled both TPU and its doped counterpart with a strain or elongation percentage of 30% to visualize how its deformation behavior reacts to when it's elongating and compressing. We found that commercial TPU had the highest tensile strength, which meant it was the most elastic and durable of the 4 samples, and additionally found that commercial TPU, when compared to its doped counterpart, required less energy to maintain its shape after being deformed, which is incredibly important to prevent any heat buildup that will deter the device in any way, shape, or form. Future research can be allocated in the creation of biosensors based on doped materials and how varying infill densities during the 3D printing process of our biomaterials affect their overall elastic modulus. Thank you.
Hello, my name is Thomas Benson, and this summer I worked in the Rehnquist lab, studying hypertension or heightened blood pressure and type 2 diabetes. My project identified hepatic endothelial cells, or blood vessel cells in the liver, as responsible for producing gamma-aminobutyric acid, GABA for short, which inhibits nerve signals to the brain. To ascertain this, my project involved collecting liver samples exposed to chemical treatments targeting 2 endothelial cell receptors, or proteins that respond to specific signals. These treatments decrease cell membrane potential, or the difference in electrical charge between the inside and outside of the cell, and stimulated GABA release. Through assay experimentation, I identified increased GABA concentrations, concluding that endothelial cells must truly be responsible for producing GABA. If they weren't, they would not have reacted to the treatments by producing more GABA. My findings contribute to Rehnquist lab research, as they disprove the previous theory that hepatocytes, or liver cells, are responsible for producing GABA. Moreover, my project holds significance because GABA is responsible for causing both hypertension and type 2 diabetes, as shown by vagotomy or nerve cut surgery. By severing the hepatic vagal nerve, which links the liver and brain, scientists eliminated symptoms of hypertension and type 2 diabetes, indicating GABA involvement. Moving forward, my lab may develop drugs that target endothelial cells, inhibiting their GABA production. Thank you for listening.
Hi, my name is Reese Bermeo, and over the summer I worked in Dr. Alicia Allen's clinical research lab, which focuses on finding ways to improve interventional treatment methods among postpartum mothers with opioid use disorder, or OUD. Over the past decade, the prevalence of parents in the United States alone suffering with OUD has increased by 600%, indicating a need for more research to improve interventional treatment methods for parents with OUD, specifically postpartum mothers. Hence, my research focus was to investigate recurring themes within the hopes and wishes of postpartum moms with OUD for their child. To begin my investigation, I did a literary search on peer-reviewed articles published within the last decade, centered around the effects of the perinatal period on the mother-infant relationship among postpartum moms with OUD. Then, I qualitatively, specifically thematically, analyzed interview data from Dr. Allen's ORCID study, which observed the relationships of caregiving activities and hormones after infant delivery among postpartum moms with OUD. The interview data that I examined was derived from study coordinators asking participants, if you had 3 wishes for your baby's future, what would it be? And what are the 3 most important things you think you can offer your baby? My analysis concluded that the 5 most prevalent recurring themes within the hopes and wishes of postpartum moms with OUD was that they hope that their baby is successful, happy, healthy, feels loved, and experiences stability in their future. The sample population that I observed was mostly in recovery for several years or more. White, non-Hispanic women, Hispanic women, publicly insured, and living on their own. By knowing the recurring themes within the hopes and wishes of these mothers, providers are able to cultivate more caring and therefore effective interventional treatment methods and support their journey to recovery. Thank you.
Hi, my name is Zachary Boydell, and I worked in the Recoy lab this summer, researching Madagascar hissing cockroach behavior in an isolated versus social situation to try and create a better foundation for future research going forward. I did this by creating 2 groups, one social and one isolated group. My social group had 6 cockroaches all in one terrarium altogether, with my isolated group having 6 terrariums with 1 cockroach each, all separated. I would observe these cockroaches for 10 minutes, 2 days a week, and I did this for 2 weeks. After those 2 weeks were over, I swapped the group's environment. So I split the social group up into isolation, and I combined the isolated group into one big social group. I did this so that I could compare both groups, and I looked at their aggressive behavior, that being ramming into another cockroach, shaking their abdomen, or using their abdomen to ram into another cockroach. I counted these and found that the social group had far higher rates of aggression than the isolated social group. This can be used to create a better understanding of these cockroaches, as it is a low-cost approach to neuroscience, which is the goal of my lab. Future research could also use the machine learning software Sleep to better understand locomotive functions in these cockroaches. Thank you.
Hello, my name is Matthew Boylan. This summer I worked in Dr. Zhu's lab, where I investigated the role of stress granule proteins in skin cells upon UV exposure. You may be wondering, what is a stress granule? Well, stress granules are a cell's natural response to stress. They consist of clusters of mRNA and other vital resources that cells sequester to maximize their chances of survival. When stress granules are unable to be disassembled by cells, they can act as a roadblock, becoming pathogenic. This is where we see the link to ALS, cancer, and other harmful diseases. Using CRISPR, I knocked out 3 key genes of interest. 1, which produces a protein which is necessary for stress granule formation, and 2 others which are closely related. We want to investigate the effects that the absence of these genes have on cell growth and stress granule development. To verify the effectiveness of our knockouts, we measure the levels of mRNA using qPCR. Then, we identify what protein is present within our cells using a Western blot. This 2-step process is necessary because in normal cells, we would expect to see mRNA and protein. In stress cells, we may only see mRNA tied up within stress granules, and in our knockouts, we want to see neither. The qPCR graph that you see shows promising levels of mRNA as compared to control. Specifically, in the Haggett skin cell knockout, we see signs of full knockouts. In the Western blot data, however, we see signs of partial or incomplete knockouts. The next step for our experiment is to isolate cells containing full knockouts to continue to grow these in hopes of achieving a full, complete knockout cell line. The cell lines produced in this experiment can be helpful for the lab to further research these genes of interest and to help produce therapeutic treatments for cancer to possibly prevent or treat it in the future. Thank you.
Hi, my name is Soleil Chung, and this summer I worked in the Brown Lab, and we investigated a G protein-coupled receptor, or a GPCR, called rhodopsin, which is found in the retina of the eye to detect light, which then activates our protein from inactive dark state to inactive meta 1 state to then active meta 2 state, allowing for dim light vision. So you may be wondering why we investigate rhodopsin instead of other GPCRs found in the body, and this is because rhodopsin's activation mechanism can be a model for hundreds of other GPCRs that are sometimes responsible for causing diseases due to their malfunction, which also makes them popular drug targets. So now knowing this, my goal for the summer was to investigate how bound water affects meta 1's light absorption using computational methods. Now to do this, we ran calculations on our meta 1 structures with anywhere from 0 water molecules to 6 water molecules surrounding our proteins. In doing this, we're able to analyze our structure's orbitals as well as the wavelength of light that they absorb, and we found that when rhodopsin is hit with light, a specific molecule inside our protein called retinal absorbs most of that light, regardless of whether it's in a hydrated or dehydrated environment. We also found that increasing the amount of water molecules surrounding our meta 1 structure not only makes our model more accurate to an experimental scenario of rhodopsin's light absorbance, but it also shifts the type of light that meta 1 absorbs to a shorter wavelength and towards active meta 2's light absorbance, which tells us that water does play a role in rhodopsin's activation mechanism. Now, all of this information here can be applied to other GPCRs to understand their activation mechanisms, which can then hopefully help us to create more effective drug treatments in the future. Thank you.
Hello, my name is Logan Curley. This summer I worked in Dr. Leroy's lab, which is a lab that focuses on the study of 2D materials, which are materials that are 1 atom thick. The material that I worked with is called graphene, which are sheets of carbon atoms derived from graphite. Specifically, I was looking at the unique electrical properties that arise when 2 sheets of graphene are stacked on top of each other and rotated slightly to form a new superstructure called a Moiré pattern. The goal of the project was to create a device that was capable of testing these unique electrical properties. To actually create the device, different materials were stacked on top of each other. The first was hexagonal boron nitride, which is used because it is atomically flat. Then the twisted graphene bilayer was placed on top of that, and then 2 graphite electrodes were placed on that to allow for conducting electricity through. Then the device was placed on a silicon dioxide chip with gold contacts to allow for easier connectivity through the graphite electrodes. For the actual testing, first a piezo force response microscope was used to take a close-up image of the twisted bilayer region to confirm the existence of the superstructure. Then the device was loaded into a cryostat where the resistivity was measured at different voltages and temperatures, one at 300 Kelvin around room temperature and one at 2 Kelvin, which is an ultra-low temperature. What this showed was relatively low resistivity values at both temperatures with a sharp peak near 0 volts. Then the device was kept at 2 Kelvin and the conductivity was measured at different voltages and in the presence of different magnetic fields. What this showed was steps in the conductivity, meaning that only certain conductivity values were possible for the bilayer. Not only that, but the values of this conductivity aligned and confirmed with other previous studies, showing that these conductivity values are produced reliably. What this means is that graphene has a potential to be used in a wide range of different technologies, including quantum computing, advanced sensing, electrical coding, biotechnology, and more. Thank you.
Hello, my name is Euzebie De Montfort, and this summer I worked in Dr. Taylor's lab, where we create new chemical reactions to better understand biological phenomenons. My project focused on modifying proteins to target and label unknown components in mitochondria permeability transition pores, or MPTPs. But for the purpose of this presentation, I'll be referring them to as mitochondrial pores. Openings of these pores lead to mitochondrial dysfunctions and eventually cell death, which remains the root cause to many cardiovascular diseases. However, the components causing these openings remain a mystery. To test our probe's efficiency at targeting and labeling specific proteins, I ran 2 trials of experiments through cell lysate. Photolabeling, which uses click chemistry to attach a reporter tag to the photoreactive groups on our probes, which will allow us to visualize any protein modification before moving on to Western blotting, so we can analyze that labeling. Although both probes contain a photoreactive group, one also contains cyclosporin A, or CSA, a molecular glue with strong affinity to the mitochondria, which will attach to cyclophilin D, a known enzyme in mitochondrial pores, before labeling those surrounding proteins. We hypothesized that we will see less labeling with our CSA-attached probe compared to its unattached counterpart, since it is directed to label specific proteins. Currently, imaging results show that our probe without the CSA attachment has a positive dosage response and is effective at labeling general but unspecific proteins in the cell lysate. However, our probe with the CSA attachment displays less but weaker signals, and this could be due to an unsuccessful click chemistry reaction or extremely low probe concentrations. In future research, we will apply similar experiments in live cells and use confocal microscopy to visualize our probe's localization to those mitochondrial pores, so we can better drug discovery advancements in creating new and more effective treatments against MPTP-related diseases. Thank you.
Hi everyone, my name is Diego del Villar, and in this summer I worked in Dr. Erica Eggers' laboratory, a lab that focuses on understanding diabetic retinopathy at the cellular level. Research has shown that in diabetic retinopathy, there are decreased outputs of Amerkin cells, which are neurons in the retina. In these Amerkin cells, there are calcium channels that are critical for signaling between neurons. Therefore, I studied if CAV 1.3 calcium channels change in GABAergic and glycinergic Amerkin cells in 6-week diabetic retinas. To do so, I looked at 10 amino-stained retina slice images, 5 diabetic, 5 non-diabetic, and quantified the area occupied by all 3 individual stains, as well as the area of CAV 1.3 calcium channels overlapping both Amerkin cells. Using ImageJ, an image analysis program, I then thresholded and analyzed each retina slice in 3 specific regions, from the ganglion cell layer to the outer plexiform layer, in the inner plexiform layer, and in the inner nuclear layer. I analyzed and graphed the areas in PRISM, a graphing and data analysis program, for all retinas in all 3 specific regions, and checked for differences using statistical testing. My research shows that there's no significant differences in the overlap areas of CAV 1.3 calcium channels in both Amerkin cells, meaning that these channels are not responsible for the decreased outputs of Amerkin cells seen in diabetes. Further research in the Eggers lab will examine this topic using electrophysiology and will look at other calcium channels to see how they affect these Amerkin cell outputs seen in diabetic retinas. Thank you.
Hello, my name is Jenny Dawn, and over the summer I worked in Dr. Barton's lab that uses optical science and biomedical engineering to design technologies for early detection of cancer. One characteristic of cancer tumors is that stiffer tumors are associated with being more malignant or invasive. My specific project aims to demonstrate the feasibility of a technique called optical coherence elastography, or OCE, to measure and visualize the stiffness of tumors, with 2 tissue groups in the experiment being soft and stiff conditioned tumors. Specifically, we placed a thin transparent silicone layer called a phantom on top of the tissue specimen and applied compressions of increasing amounts of axial force. The resulting deformation or strain of the phantom would provide relative measurements of tissue stiffness, since stiffer tissue regions would press on the phantom more and make it more deformed or thinner. We used an imaging system called OCT to produce cross-sectional image stacks so that we were able to see and measure the thickness of the phantom at different points along the image. These data points were converted to a matrix of phantom strain values corresponding to the original image that were then converted to a color map using the software MATLAB. By overlaying the color map onto the original top-down image of the tissue, we are able to identify stiff and soft tissue regions, and the maps show an overall higher stiffness in the stiff conditioned tumor than the soft conditioned tumor. Our results demonstrate the feasibility of using OCE to identify stiff tumor regions. In a clinical setting, tissue stiffness can act as an indicator for potentially cancerous areas. OCE can be implemented in endoscopes and other minimally invasive diagnostic strategies to further characterize regions of interest and improve accuracy and early detection of cancer. Thank you.
Hi, my name is Ryan, and this summer I worked in the Song Lab where they study cell platform technologies to study how cells respond to their surroundings. My project specifically was optimizing a hydrogel to better mimic the extracellular matrix, or ECM, in terms of chemical structure, topography, and conductivity. To use this, I used 4 materials. First was PEGDA, which is a blank slate base hydrogel which cures in the presence of a crosslinker. Second was gelatin, a degraded form of collagen, one of the primary proteins found inside of the ECM, so we used it to better mimic the chemical structures inside of the ECM. Third was PDOT, which is a biocompatible conductive polymer which induces its property of conductivity once implemented into the gel solution. And finally was potassium persulfate, which is the chemical cross-linking reagent which turns the liquid solution into a solid gel by making bonds in between the individual molecules of the different materials. These were the methods we used. We used soft lithography on a silicon wafer by mask and UVing it to make 1 to 10 sized micro patterns on the surface of the silicon wafer. Then we heat cured the hydrogel solution on top of it so that once we peeled the hydrogel off of the silicon wafer, we would see a similar topography. At the top right, you can see figures A and B are examples of the silicon wafer with the patterns on it, and the other 4 images are examples of the complementary gels that were peeled off, both non-conductive and conductive. To optimize conductivity, we found that 45% volume was the best concentration of PDOT to use, and any concentrations higher than that resulted in an inhomogeneous solution, non-cross-linked gel layers, and non-conductivity. In the future, we hope to seed neural cells on top of the hydrogel to engineer in vitro neurons to help with cell adhesion and neuron alignment to counter debilitating spinal cord injuries or other nervous damage. Thank you.
Hello, my name is Camila Garcia Amezquita, and this summer in Dr. Sutphen's lab, I studied how pre-starvation conditions affected breast cancer cells' reaction towards tamoxifen, a type of breast cancer treatment. Our goal was to see if pre-starvation conditions could sensitize breast cancer cells towards tamoxifen while making normal cells resistant towards the treatment. We did this by using C. elegans, a type of microscopic worm, represent normal cells, since they share many genetic similarities with humans, including FOXA protein, which is my protein of interest since it regulates cell cycle and cell death. In breast cancer cells, we measured the gamma H2X gene, a marker for DNA damage, and oxidative stress levels, because fasting should increase damage towards the cell. In C. elegans, we measured FOXO activation levels to then analyze how stress mechanisms reacted towards radiation. Using breast cancer cells, we did immunofluorescence staining of the gamma H2X gene. Cells were then treated to 4 different conditions, including starvation followed by tamoxifen treatment. Our data showed that the gamma H2X gene was found most in starved cells treated with tamoxifen than any other condition, suggesting that starved cells did become more vulnerable towards the treatment. Additionally, oxidative stress levels were elevated within starved cells, further proving our hypothesis. DAF16 C. elegans exhibited higher FOXO activation under radiation conditions, demonstrated by lower immunofluorescence. Increased FOXO activation led to an increase of SOD3P activation. SOD3P activates raw scavengers to then get rid of superradicals and repair DNA damage. With these results, we can conclude that DAF16 FOXO plays a protective role in C. elegans, while in breast cancer cells, starvation does enhance sensitivity towards tamoxifen. This research can be used to advance future cancer therapy strategies by uncovering how cellular response to stress can be used to advance cancer treatment. Thank you.
Hello, my name is Sophia Glenn, and I worked in the Duckworth lab researching the reproductive success of zebra finches. So our question was, does female choice impact compatibility, and does this influence reproductive success? We had 2 groups, females that could choose their mate and females that were picked with the mate. We focused on key behaviors such as allopreening, which is affectionate preening, social feeding, and mirroring, also known as behavioral synchrony in birds. We discovered that in our results, while birds who were more affectionate, meaning compatibility, were closer with each other, that did not result in reproductive success. The pairs that were less affectionate but were more focused on nest building and parental care were higher in reproductive success and were more likely to mate than those who were more affectionate. One thing that was not considered in this study was male choice and male age, which would most likely impact compatibility in reproductive success by having their age impact their experience on mating. Their experience and age plays a large role in how they interact with the female and how they interact with their offspring. This research will help conservation efforts in endangered species. Thank you.
Hello, my name is Stephy Hein, and over the summer I worked in Dr. Bao's neuroscience lab, where we study how hearing loss can change the brain in ways that lead to cognitive disorders. What I focused on specifically is how hearing loss impacts the amygdala, or the part of the brain that detects fear. There are 2 main components to my experiment. First, we studied the behavioral changes that occur as a result of hearing loss, and then we studied those changes at the cellular level. For the behavioral changes, we performed what is called an open field test, where we placed several mice into separate containers and then tracked their movements. What we found is that after hearing loss, the mice tended to prefer corners. This preference towards corners is often associated with fear and anxiety. So from that test, we can conclude that hearing loss does in fact make mice exhibit more fear and more anxiety. Now moving on to the cellular portion of my experiment. We studied brain scans before and after noise exposure occurred, and we took a focus on immune cells in the brain. The reason we focused on immune cells is because immune cell hyperactivation is often seen in cognitive disorders. And that's exactly what we observed. After noise exposure, immune cells grew larger not only in size, but also they became more inflamed, indicating a hyperactive state. So from that, we can conclude that immune cell hyperactivation occurs after hearing loss, which can lead to the behaviors that we observed in the first part of our experiment. In future research, we hope to target that immune cell activation, perhaps preventing it or even curing it, which can lead to a cure of the cognitive disorders of fear and anxiety. Thank you so much.
Hello, my name is Kristy Hudgins, and over the summer I worked in Dr. Gulabev's lab, which focuses on modeling atomic physics using computer methods. My project in the lab focused on finding a way to model high harmonic generation, or HHG, in a way that students can better understand and visualize the process. HHG is the process in which a laser is first shot at a gas. The electrons inside the gas have their potential energy start to oscillate. This allows the electron to first ionize or leave, and then subsequently recombine or return to the atom. When this happens, the excess energy gained from acceleration is emitted as a UV photon. This helps us visualize electron dynamics. In our simulation, we first started off by graphing the laser field and the corresponding potential energy of the electron. Then, using equations like the displacement equation on the left, we added more graphs, like displacement over time, velocity over time, and two phase diagrams, which are velocity over displacement and kinetic energy over displacement. Using these graphs, students can better visualize the electron trajectory at different ionization times. The user of this interface can decide at what time the electron should be ionized, and this determines a whole bunch of factors like the recombination time, the maximum displacement, and the velocity. However, because we're using classical physics equations and trying to model a quantum process, we would need to use quantum equations to create a better model of HHG. Nonetheless, students can still use the simulation to see how the electron trajectory changes based on different ionization times. Thank you.
Hello. Imagine a future where farmers are able to predict the productivity of their plants just by analyzing the air around them. As climate change and food insecurity become a larger question in today's society, this research hopefully aims to address those questions. This summer, I interned at Dr. Meredith's lab, where I study volatile organic compounds known as VOCs, which are chemicals that easily evaporate into the air. VOCs are emitted by various parts of the plants, from the roots, stem, and leaves. My research focused on the VOCs emitted by the roots of 5 sorghum genotypes. My hypothesis was that more productive sorghum will display a higher concentration of diverse VOCs over time. To measure the VOCs, an underground soil probe is placed underneath the sorghum roots. The sample is then collected and purified using ultra-zero air to ensure no excess moisture. The purified sample makes its way to the Vocus, a machine that measures VOCs to be ionized for further analysis. The Vocus's high sensitivity allows for the VOCs to be sorted by polarity, accurately distinguishing the VOCs. The Vocus is also able to measure the VOCs in real time, and the data is analyzed to study the relationship with plant productivity and VOC concentrations. Although my current research is still in progress, prior studies have shown the direct relationship with plant productivity and VOC concentration. Some limitations of my current research involve the growth stage of the sorghum roots. Since the sorghum roots are still not yet mature, it is difficult to see the trend between VOC concentration and plant productivity. However, current research indicates a positive trend for more productive plants in the day display a higher concentration of VOCs over time. Sorghum genotypes usually display 5 major VOCs, additionally with nitrogen-based fertilizers introducing new ones. Future research will aim to identify more diverse VOCs from more sorghum genotypes, enhancing our understanding of plant productivity. Thank you.
Hello, my name is Eugene Jap, and this summer I worked at the Snyder Lab. We are researching the ceramide pathway, which is involved in cell messaging throughout the body and is impacted by a dietary fat intake. Our goal is to understand how different high-fat diets and loss of key enzymes, specifically ceramide synthase 5 and ceramide synthase 6, which I will refer to as CERS 5 and 6, impacts sphingolipid generation in the colon. Our model has a knockout or loss of SIRS-5 and 6 within villi cells, which are found within the small intestines and the colon. We specifically look at SIRS-5 and 6 because these enzymes generate ceramides with carbon chain lengths of 14 and 16, which has been known to cause inflammation. I extracted lipids from colonal tissue after the 3 diets, one being a low-fat or controlled diet and 2 high-fat diets, one with milk fat and the other with lard, and then these lipids were analyzed using mass spectrometry. Through this, we found that lard-fed females with a loss of SIRS-5 and 6 had the greatest change in sphingolipid generation, while the other diets did not see any significant change. The findings of this research will contribute to the emerging field of lipid cell messaging and help us better understand how commonly consumed foods regulate biological functions. Thank you.
Hi, my name is Katie Jennings, and this summer I worked in Dr. Cheret's lab. The lab focuses on cell migration, which is important as it relates to cancer metastasis. Metastasis is the spread of cancerous cells away from their original tumor site, and more specifically, the lab looks at one protein complex, mTORC2, which is often responsible for cell migration. My project focuses on an additional 3 proteins, PI3K, AKT, and RAS, which are all nearby to mTORC2 in signaling pathways. I sought to determine the relationship between the 2 pathways shown in blue and green. I inhibited PI3K, outlined in green, and left the RAS mTORC2 pathway in blue. I utilized 2 cell lines. The first are empty hygros, a control line, and the second, a wild type, which has an overexpression of RAS. I was able to determine this relationship as each of these pathways adds a unique phosphate marker onto AKT, producing a phosphorylated AKT. I used that in my data analysis to create a ratio of phosphorylated AKT at each of those sites over total AKT. For my results, I gained 4 graphs. Those marked in blue at the top, we expected to remain constant from the 0 nanomolar point, as RAS and mTORC2 were not inhibited in this experiment, and mTORC2 is this site's responsible protein, but there was no identifiable trend. Those marked in green we expected to decrease from the 0 nanomolar point, as PI3K is the responsible protein for the site, and we inhibited it. But again, there was no identifiable trend. Our results are due to not having a sufficient inhibitor for PI3K. So in the future, the lab will do reruns of this experiment with an effective inhibitor for PI3K, as well as utilizing additional cell lines and repeats. Thank you.
Hello everybody, my name is Maximo Jimenez Lugo, and this summer I worked in Dr. Kim's prosthetics lab, which focused on helping veterans who needed prosthetics. So as for veterans who need prosthetics, an issue that they face is that prosthetics such as percutaneous osseointegrated prosthetics, which are implanted straight into the bone, aren't too compatible with human skin as they should be. This leads to skin degeneration, infection, and if the infection is bad enough, possible loss of the entire bone. A solution that our lab came up with was keratin-coated prosthetics. Keratin is a protein that is found in hair, nails, skin, and wool. Our thought process behind this was that since skin and nails bind very well together, although they have different mechanical properties, our thought process was, what if we coat prosthetics in keratin? My role in the lab was to synthesize the keratin so we could use it. And as for my ability to do so, I was successful. As you can see on the STS page gel, the molecular weight matches around 51 kilodaltons, which is the exact weight of the keratin protein. And as for the graphs, we did this for 3 batches. As for the first graph, in the 3rd batch, the concentration was extremely low, so we had to do protein concentration. And as you can see in the 2nd graph, in the 3rd batch, the protein concentration went up by 6 times. This keratin will be used in further tests to prove if keratin-coated prosthetics will prevent the foreign body reaction so we can save people's limbs. Thank you.
Hi, my name is Adhip Justhy, and this summer I worked in Dr. Witty's experimental ultrasound and neural imaging lab. Many neurological disorders, such as epilepsy or Parkinson's, are linked to abnormal brain activity. However, modern non-invasive imaging technologies, such as EEG or fMRI, are spatially and temporally limited in providing proper visualization and localization of this activity. This incites a need for non-invasive imaging modalities to map the brain. The Witty Lab is developing transcranial acoustoelectric brain imaging, or TABI, which uses ultrasound to induce localized electrical changes for high-resolution electrical mapping. To study the TABI modality, we use head models or head phantoms. Generally, the lab uses a simple conductive agarose model. However, I was tasked with developing a multi-layer model meant to emulate the brain properties of gray and white matter layers of the brain. To study the effectiveness of this material for TABI imaging, we ran a 6-contact electrode through the gray and white matter, stimulated the electrode, and used ultrasound to make electrical maps. What we found was that the acousto-electric signal was successfully detected in both the gray and white matter. However, the signal was significantly stronger in the gray matter than the white matter, most likely attributed to its acoustic properties. These results indicate to us that the gray and white matter multi-layer phantoms are viable models for future TABI studies, especially multi-layer TABI studies, which can be incredibly helpful for the future of non-invasive brain imaging. Thank you.
Hello everyone, I'm Nishita Karikatti, and this summer I worked in Dr. Alicia Allen's lab. Opioid use disorder, or OUD, is one of the most prevalent and hardest substance use disorders to overcome, with relapse rates about 30 to 70%. As you guys can see in the map, Arizona is one of the most vulnerable states for OUD, which is why the ORCID study comes into play. The ORCID study's main goal was to focus on observing the relationship between hormones and caregiving activities after infant delivery. There are 3 parts to the ORCID study. The first part was blood sampling, which is when a blood sample was collected from the participant and then separated out for plasma, and that plasma is sent for hormone analysis. The second part was saliva sampling, which is when a saliva sample was collected from the mom and analyzed for specific hormones such as cortisol, which is a stress hormone, and oxytocin, which is a love bonding hormone. The last part was the surveys, and this is the part that I mainly focused on. Surveys were taken electronically on a weekly basis at the convenience of the respondent. My sample population that I mainly looked at were Hispanic women who lived in Phoenix, had some sort of college education, were on current medication treatment for OUD, and were publicly insured, and were several years within recovery. I qualitatively, specifically thematically analyzed their responses for week 1 and found that the most recurring themes were that mothers like to spend time with their baby, think about how far along they are on their journey, find some sort of distraction or try to stay busy, and talk with their support groups. Some future research that could be done is analyzing the change of coping mechanisms over time from week 1, which is what I did, and month 5, which is the end of the ORCID study. Additionally, identifying a causal relationship between a specific drug and a specific craving will extensively improve on existing interventions. Thank you.
Hi, my name is Braydon Kim, and this summer I worked in the Kai lab. In this lab, we focus on a neuron of interest called the PKC delta neuron. These neurons are important because when they are activated, they cause a suppression of eating behavior. So for my project specifically, I focus on a region called the central amygdala, and this region is important for the control of eating, and I also focused on amphetamines and how they affected the PKC delta neurons located in the central amygdala. For our experiment, we had 2 groups of mice. We had one that was exposed to saline and one that was exposed to amphetamine. We used immunofluorescence and imaging process to analyze how these neurons were activated in both mice groups. Looking at our results, we saw that amphetamine had a higher activation as shown with the red dots. These red dots represent CFOS. CFOS is a neural marker that we use to determine whether these neurons are actually being activated. Compared with saline, we see that there is a lack of red dots and that there was only the representation of the PKC delta neurons, which are represented by the green dots. So our results demonstrate that amphetamines cause a general activation of the PKC delta neurons in the central amygdala. However, one problem with our results is that our neurons of interest are not specifically targeted by amphetamines. As shown with the pictures, the difference between red dots and orange dots. These orange dots represent our specific PKC delta neurons are being activated compared with the red dots, which represent other neurons being activated. Therefore, in order to improve our experiment, we want to provide chemogenetics and optogenetics in order to specifically target our neurons of interest to see if they are affected specifically by amphetamines. Our results are also important in order to further understand the role of the central amygdala in the eating process and also understand how to specify targets for treatments for obesity, Prader-Willi syndrome, among other eating disorders. Thank you.
Hi, I'm Danny, and over the summer I worked in Dr. Koshi's lab studying the parasite Toxoplasma gondii. Toxo is an intercellular parasite that infects around 1/3 of the population, but normally it's asymptomatic. When it does show symptoms, it's things such as brain inflammation and increased risk of neurodegenerative diseases. Toxo forms a chronic infection by switching between 2 forms, tachyzoites, which are a quickly replicating stage, and bradyzoites, which are an insisting latent stage. The bradyzoite stage is what allows it to stay in the brain for long periods of time and emerge at a moment of immune suppression, where it can actually harm the body. The Koshi lab specifically is studying the bradyzoite stage and has found a gene, TLO, or the little one, that we believe is important to the maintaining of these cysts over time. So with this knowledge, the lab created 3 strains: a wild type, a knockout, and a complement. We infected around 35 mice with each of these strains, and then we removed their brains and sectioned them to see the difference. We stained using immunohistochemistry, staining for EBO1 antigens, which simply stains for microglia and macrophages, to measure the immune response of the mouse. At 3 weeks post-infection, both the wild and the knockout were equal, but at 8 weeks post-infection, while the wild remained constant, we saw a significant decrease in the knockout. This aligns with previous data found in the Koshi lab, showing that the cyst count also decreased over 8 weeks. This leads us to believe that the gene TLO has an important role in maintaining these cysts over time. Future research that we could do would be infection over a longer period of time, or staining for T cells as well, to see if those align with the results we got. There's no cure for toxoplasmosis, which is why this could be an important stepping stone into getting there. Thank you.
Hi, my name is Annika Kessler, and this summer I worked in Dr. Hassan's atomicroscopy lab. What is atomicroscopy? Simply put, this is a way to use ultra-fast laser pulses to measure electron movement in certain materials. Now, why would we need ultra-short laser pulses, and why are they important? They're important for 3 reasons. The first reason is to be able to image electron movement, you must have frames of time that are shorter than the entire orbit of an electron around the nucleus of an atom. The second reason is because of the spatiotemporal overlap, or simply put, having the laser pulse in the right place in the right time. This also relates to the third reason, which is the equation distance equals the speed of light times time, which basically shows the relationship between space and time. This means that any change in the position will result in a change in time, or any change in the length of an ultra-short laser pulse will result in a change in the distance. This is shown in the graphs on the right. So you can see in the first graph that when the laser pulses are perfectly aligned, that they will combine and form the shortest possible laser pulse. However, in the second graph on the right, they're misaligned and they're not overlapping in both space and time, which means they will produce a laser pulse that is not optimal for imaging electrons because it is not as short as possible. My research in this lab focused on using a method called frequency-resolved optical gating, or FROG, to measure the temporal duration of ultra-short laser pulses. The first part, frequency-resolved, focuses on the fact that the data that we can achieve from this method measures the frequency, or color, of light that is emitted. The second part, optical gating, refers to the fact that we can use optical techniques to manipulate lasers in order to have them overlap in both space and time. Using the FROG method, I was able to determine that the length of one ultra-short laser pulse produced by our lab was 5 femtoseconds, which is a precision of a few quadrillionths of a second, or to put it in perspective, a few millionths of a billionth of a second. This data that I produce shows that FROG is an optimal technique to characterize the temporal and spectral components of ultra-short laser pulses, and can be utilized in a variety of optical techniques. In the future, FROG can be used by my lab to determine the spectral and temporal components of ultra-short laser pulses and hopefully improve atomicroscopy and electron imaging technologies in the future. Thank you.
Understanding the role of certain non-coding RNA is crucial in understanding Ewing sarcoma, an aggressive type of bone cancer mainly diagnosed in kids and teenagers. My name is Benite Luhando, and this summer I've been working in Dr. Schwartz's lab. studying the role of cytoskeleton regulator RNA in Ewing sarcoma, a genetic material that does not code for protein but plays a crucial role in cell regulation, cancer development, and HIV latency. My lab mainly focused on finding the presence of this RNA in Ewing sarcoma. To test our hypothesis, we cultured cells, isolated RNA, carried out a cDNA synthesis, and conducted a qPCR. Our results suggested that our cells grew by 83% viability after culturing the cells, which was approximately 17% of our cells had died, but leaving us with enough cells to continue with RNA isolation. Our RNA sample full spectra showed that our S670 wild-type cells had a higher concentration than the jacket cells, which suggests that there might have been some impurities like DNA. So we did our cDNA synthesis, which we were able to get enough cDNA to continue with further research. We conducted a qPCR, and our results suggested that there was amplifications of all the relevant primers that we used, confirming the presence of cytoskeleton regulator RNA in Ewing sarcoma. Future research will delve deeper into the role of this RNA in bone cancer. Thank you.
Hello, my name is Julia Meindl, and over the summer I worked in Dr. Brown's chemistry and biochemistry lab. Now, do you know what anxiety, type 2 diabetes, and migraine medicine all have in common? So they all target something called G protein coupled receptors, or GPCR, which my lab is looking into to further understand how they function. We're using a very specific one called rhodopsin. Now, rhodopsin is really cool because it's found on the retina of your eye and contains a light-activated molecule called 11-cis-retinol. And when the retinol absorbs enough light, it starts to send a wave of signals that eventually give us dim light vision. My specific project was looking into how water molecules interact with the retinol before it's absorbed enough light to actually be activated. So to do this, we isolated the retinol, then added in 10 amino acid residues around it, as well as 1 to 7 water molecules to create 8 trials total. Then, for each trial, we would optimize the structure, meaning that it's at the lowest possible energy so that way the structure wouldn't change, and then we ran a second series of calculations to get 2 different types of results. So the first type of results that we got was the molecular orbitals of the structure. Orbitals are where electrons of a structure reside, and when they're hit with enough energy, in this case provided by the light, they can jump from the highest occupied orbital to the lowest unoccupied orbital, and the gap in between them can actually determine at what wavelength the molecule will start to absorb light. Instead of using that for the calculation, however, we also got something called a UV visible spectrum, which is a graph that shows us at what wavelength of light the most light was absorbed by a molecule. So we found that all trials absorbed at about 500 nanometers with their tallest peak, and trials with 4 or more water molecules also had a slightly smaller peak at 380 nanometers. The final calculation that we did is we took a difference. So we had an experimentally calculated value of 498 nanometers and subtracted each calculated value from our trials from that to create a difference graph. From that, we found that a structure with 6 water molecules is most similar to an experimentally determined structure, which is useful for anyone who wants to further investigate with rhodopsin. This information can be applied to the over 800 GPCRs that are related to rhodopsin, and this information can be sent to drug companies to create more effective drugs targeting rhodopsin's family. Thank you.
Hello, my name is Brandon Mokhtarian, and this past summer I've been working in the Giannetti lab. During my time in the Giannetti lab, my research has been centered around designing a photocatalyst. And for those who don't know, a photocatalyst is something that speeds up the rate of a photochemical reaction, and its uses range from solar panels to water filters. The photocatalyst that I was designing is special and different from the industry standard because not only is it far less toxic and more environmentally friendly, but it's far more versatile and easily modifiable, meaning we can tailor it to specific reactions, whereas other photocatalysts take a more one-size-fits-all approach. Another special feature of our catalyst is that it combines the photoredox catalytic cycle with a transition metal catalytic cycle, which makes it far more efficient than others as well. And to synthesize this photocatalyst, which is labeled as number 4, the first step we must take is synthesizing the pendant arm, which allows us to later bind metals to the photocatalyst. That reaction is the one labeled on the top, and once we make the pendant arm, we can attach it to the molecule labeled number 2, which is called TLG2, and once we attach the pendant arm to the TLG2, we can create our photocatalyst. And then the final step would be binding a metal.
As you can see from this NMR spectra, which basically confirms or denies the structure of a molecule, we can see that we were successfully able to synthesize the pendant arm. And based on the second NMR reading on the slide, we were also able to successfully synthesize the photocatalyst minus the metal. However, we did have some difficulty in purifying this, which can be seen in the large peak in the center. Thank you.
Hi, I'm Jishnu Nayak, and during the summer I worked in the Largent Milnes's lab, which focuses on medication overuse headache. Migraines are one of the most common neurological disorders, and they're treated using medications such as triptans, which work by binding to serotonin receptors and constricting blood vessels in the brain and preventing the release of neuroinflammatory peptides. However, overuse of these medications can lead to medication overuse headaches, creating a vicious cycle. So my focus was to study a migraine medication called sumatriptan, building off of the results of a prior experiment which analyzed medication overuse headache sex differences. In that experiment, females had more medication overuse headache, which is why in this experiment, we implanted several concentrations of sumatriptan into male rats. And what we ended up finding was that the magnitude of allodynia, which is a symptom of medication overuse headache that involves pain to non-painful sensations, was higher in certain concentrations. Allodynia is an indicator of medication overuse headache. And what we found was that 3.4 milligrams of kilograms of sumatriptan and 0.6 milligrams of kilograms of sumatriptan had the highest magnitude of allodynia and thus medication overuse headache. And this is also corroborated by another graph which shows the population of the rats that had allodynia. We also ran a Western blot and looked at the neuroinflammatory profile of rats of a prior experiment and found that CD68, a protein which is found in macrophages and thus represents inflammation, was expressed in greater amounts in the rats that had sumatriptan. The future directions for this experiment involve preventing and reversing medication overuse headache through inhibiting the degradation of endocannabinoids, which are molecules that regulate pain in the nervous system. It also involves testing other migraine medications, such as other triptans and opioids, and also continuing the Western blot to analyze the neuroinflammatory profile of the rats that were used in this experiment, so the male rats. Thank you.
Hi, my name is Pask Nelson, and this summer I worked in Dr. Daniel Pappage's insect ecology lab, studying the interactions between insects and plants. My project focused on how Palo Verde colors change over time and whether bees can perceive this change. I studied flowers from two species of Palo Verde, the Mexican Palo Verde and the Desert Museum hybrid. An important part of the flower's anatomy is the banner petal, which is a prominent petal at the top of the flower that attracts bees to the flower while they're foraging. Bees can see UV light, which means they perceive color differently than humans do. To measure the flower colors, we measured the wavelength of light reflecting off of the flowers, and then we plotted these colors in bee color space to tell whether bees can distinguish between flowers. We found that there's a clear difference between new and old flowers in both species, which is seen in changes to the banner petal. In the Desert Museum hybrid, the old banner petals have a lower reflectance at long wavelengths than the new banner petals, while in the Mexican Palo Verde, the old banner petals have a higher reflectance at long wavelengths than the new. Both of these changes are distinguishable by bees, as seen in the bee color space plots, as a noticeable difference between the points for new and old banner. Being able to distinguish between new and old flowers benefits bees because they can collect more pollen and nectar by avoiding old flowers, which likely have less of these rewards. This information can help in future research in conservation and agriculture. Thank you.
Hello, I'm Tara Nukla. Over the summer, I worked in Dr. Slipfin's lab studying the molecular and cellular processes of aging. Aging is important to understand due to the prevalence of age-related diseases worldwide. Our experiment is built off of a previous experiment that stated an increase in dietary cholesterol increased the lifespan of C. elegans, a type of microscopic worm. We also realized that when blocking cholesterol's ability to act as a signaling molecule, this increase doesn't occur. Therefore, we concluded that cholesterol's ability to signal is vital in the process of aging. We aim to further understand this by understanding where cholesterol is trafficked throughout the bodies of these worms. We realized that we had to use two different living conditions, petri dishes and a worm motel, which is a silicone dish. We did this so we could understand the difference between the living conditions for future experiments. We then used two different techniques to image the worms. We first fed them fluorescent cholesterol to image them, and then we used oil red O staining to image the lipids. This is important because cholesterol is a signaling molecule in the mTOR pathway, which regulates lipid growth. We realized that worms in the cholesterol group accumulated more cholesterol as they aged, and they accumulated it in specific areas of their body. We also realized through the oil red O staining that they had more highly ordered areas of lipids in the cholesterol group. We aim to further this research to delay or even reduce the onset of age-related diseases in humans. Thank you.
Hello, my name is Mallory O'Brien, and this summer I worked in Dr. Gall's lab studying the effects of nutrient deprivation on the levels of NRF1. Cardiovascular disease kills almost 18 million people around the world annually. Now, the protein NRF1 has been linked to a variety of heart health benefits, including protecting heart cells from dying, replenishing ones that do, and even reducing tissue scarring in the heart. A recent research study from my lab has shown that starvation can increase the RNA levels of NRF1 in rat cells. The research project I worked on aimed to answer the question of whether nutrient deprivation can increase the levels of the protein NRF1 for increased cardioprotection. To do this, we first cultured and starved a rat cell line and then conducted a Western blot, a lab technique to visualize the band intensity of certain proteins, where the thicker and more intense a band is, the more of that protein there is. Two important parts of this process were distinguishing between the inactive and active sizes of NRF1 and using the gene GAPDH to account for any errors in loading the samples. Our samples included a 17, 16, 8, and 2-day starved sample, as well as a GROW sample. The GROW sample was set to 1 in the data, just to easily compare the other samples to each other. We observed bands at 3 different sizes, 95, 46, and 25. As you can see, for the most part, the 46 size samples followed the expected trend of increased band intensity and therefore protein levels for increased starvation period, while the 95 and 25 did not. Future work with this project will be looking at similar starvation conditions with heart cells. These results are important because understanding the specific cellular conditions in which NRF1 is activated, as well as the sizes of NRF1 activated, are vital for creating future treatments combating cardiovascular disease using the activation of NRF1. Thank you.
Hello, my name is Alec Peral, and this summer I worked in Dr. Kenneth Weiland's lab. My role in this lab was to work with hemodialysis patients who are at risk of developing cardiovascular diseases due to their poor diets and increased intake of sodium. So before starting the development of our program, we developed some inclusion and exclusion criteria. Some of the inclusion criteria included them going to dialysis 3 times a week, them being over the age of 18, and being on dialysis for over 3 months. Some of the exclusion criteria are if they are already on a low sodium diet, if they have sensory or physical limitations, and if they experience epilepsy or vertigo. So to work with our patients, we developed an innovative and comprehensive 3-step strategy called Grow, Shop, Cook. So Grow, our idea was to develop a garden outside of dialysis clinics where patients can go and pick fresh herbs to use at home. Shop is a 6-lesson virtual reality simulation where patients will be taken around a grocery store and taught how to properly shop. And Cook is a nutritional lesson and a cooking class that patients will receive over an 8-week span. We hope that using these innovative strategies, we are able to better the diets of patients and lead to a better quality of life. Thank you.
Hello, I'm Mateo Pimienta, and this summer I worked in Dr. Babus Costeca's plant science lab. One focus of our lab's research is on contaminated mine sites and how plants native to the desert southwest can potentially be used to reabsorb these contaminants through their roots and remediate these polluted ecosystems. In our experiment, we used 2 plant species, Atroplex lentiformis and Atroplex canescens, both native to the desert southwest, along with the heavy metal copper as a contaminant, which can be toxic to plants in high concentrations. In our experiment, we grew these plants over the course of 7 weeks, hydroponically, slowly increasing the concentration of copper solution that we administered to them. In our experiment, we used 3 plant groups, 2 which were grown from commercial seeds, and another group whose seeds were found growing near the Iron King mine site in northern Arizona, so they have been previously exposed to heavy metals. After the 7 weeks, we found that none of the plants in the treated groups grew over 7 micromolars of copper solution. However, the control group, which was kept at a constant 0.1 micromolars of copper, was able to grow throughout the 7 weeks. Furthermore, when we weighed these plants, we found that the biomass of 2 of the control groups was greater in both the leaf and total biomass compared to their respective treatment groups. Moreover, we found that for all 3 control groups, the amount of leaf as a percentage of the total biomass of the plant was greater for all 3 control groups compared to the treatment groups. This shows that while high copper exposure does hinder the growth of Atriplex, these plants can still tolerate copper to an extent, so further research needs to be done to find in what situations these plants can be planted at these mine sites. And further experiments will be done to narrow down and identify the specific copper tolerance of Atriplex so that we could use these plants to remediate these polluted areas. Thank you.
Hello, my name is Evelyn Pombo and I interned in the Maudevon lab this summer. Parkinson's disease is a neurodegenerative disease characterized by progressive motor and neurocognitive decline over the course of aging, and also by the loss of dopamine-producing neurons in a motor region of the brain called the substantia nigra. Currently, no treatment for Parkinson's disease exists that is capable of stopping or even slowing the progression of the disease. In the brain cells of those with Parkinson's disease, we see the accumulation of Lewy bodies, which are basically clumps of a protein called alpha-synuclein that can misfold and aggregate to cause cellular toxicity. Also in these cells, we see significant mitochondrial dysfunction, which brings me to the drug we're testing, lasmiditan. Lasmiditan is an FDA-approved migraine medication that our previous data found can induce mitochondrial biogenesis and reduce Parkinson's disease pathology and symptoms in young mice at an earlier stage of the disease. Presently, we tested whether or not it could do the same thing, induce mitochondrial biogenesis or the creation of new mitochondria, but this time in an older, aged alpha-synuclein overexpressing mouse model of Parkinson's disease, aged 10 to 12 months. Our molecules of interest were PGC1 alpha, the master regulator of mitochondrial biogenesis, alpha-synuclein, and mitochondrial DNA. And our regions of interest were the cortex, the striatum, the substantia nigra, and the hippocampus. So we found that there is a trend towards lasmiditan treatment rescuing the PGC1 alpha phenotype, particularly in the striatum and the cortex, though it's not significant with this small of a sample size. Lasmiditan treatment had no effect on alpha-synuclein in any region, and then finally, lasmiditan treatment significantly increased mitochondrial DNA in the cortex, with an increasing trend noted in the substantia nigra. Overall, with this preliminary data, we can assume, or this preliminary data suggests, that lasmiditan might be more effective in younger mice at earlier stages of Parkinson's disease than in older mice with advanced Parkinson's. Thank you to the Maudevon Lab and the KEYS internship program.
Hello, my name is Miranda Quiñones, and this summer I interned in the Limison Lab, which focuses on radiotherapy-induced collateral damage to the salivary glands, more specifically the parotid gland, as it's the most radiosensitive. Previous research found that the response to radiotherapy in the parotid gland was an increase in nuclear YAP, which is a protein that controls the multiplication and division of your cells. It was also found that there was disruption made to the cytoskeletal structure of your salivary gland cells. These both contributed to a loss in salivary flow, dry mouth, and an overall decrease in the quality of life. To test the role of YAP in the dysfunction process, 2 group models were made, one expressing YAP and one with YAP knocked out. The group with YAP knocked out, even prior to radiation, had a significant decrease in salivary flow. This led us to the question, is YAP also responsible for the changes made in the cytoskeletal structure of salivary gland cells? To test this, I decided to measure the cytoskeletal area amongst both groups. I hypothesized that if there was a significant difference in the cytoskeletal area, this would mean that YAP regulates the cytoskeleton and is a possible pathway for dysfunction. This is because, in a functional model of the salivary gland cell, the cytoskeleton surrounds vesicles of salivary protein, allowing for salivary protein to be transported into saliva. In a dysfunctional model, the cytoskeleton clumps together, leading to a decrease in area and prohibiting the transport of salivary protein. To test this, I decided to use a technique called phalloidin staining, which is a fluorescent stain that binds to the actin in the cytoskeleton, allowing me to measure and compare the area of both cytoskeletal tissues. From my results, I found that there was no significant difference in the cytoskeletal area, meaning that YAP does not regulate the cytoskeleton. From this, we are able to turn our attention to other possible pathways of dysfunction in hopes of reaching our goal of creating long-term treatment options to reverse chronic damage done to the salivary glands. Thank you.
Hi everyone, my name is Alina Rafiath and I was working in Dr. Karn's pharmacogenetics lab where they look at how genetic changes affect patient drug response in cardiovascular medicine. So inaccurate dosing often causes adverse effects like excess bleeding. My project to improve accurate dosing for a specific blood thinner called warfarin is looking at copy number variations, which are deletions in DNA that affects a varied number of copies between individuals. And we're looking at copy number variations on our gene of interest called PHIP and detecting whether those are associated with warfarin dose requirement in the Hispanic Latino population. We chose copy number variations because they're becoming a larger focus in this field, and single nucleotide changes in DNA have already been well studied. Copy number variations are also, they affect a larger portion of DNA, so they have a bigger impact to look at. Our methods revolve around quantitative PCR, where we amplify a gene of interest as well as a control gene, and then we get light signals from those. After running the light signals, we analyze the light signals to get the number of copies each sample has of our gene. We then look at the clinical database to get the dose each patient has of warfarin and compare that to the copy number of the gene they have. We then did regression line association, and after running the association, we found no association between copy number on PHIP, our gene of interest, and warfarin dosing in the Hispanic population. However, this study does contribute to the candidate approach of using genetics to find precision medicine. In the future, we know to use other methods to find associations, as in targeted DNA sequencing. Thank you.
Hi, my name is Sadie Schwartz and I interned in the Duca lab over the summer. Dr. Duca and his lab are primarily focused on gut and brain signaling and the factors that affect it. They, along with many others, have determined that gut peptides play a crucial role in the gut's communication with the brain, along with regulating food intake, weight gain, and glucose levels. My job this summer was to examine a specific gut peptide called CCK and observe how it activates neurons in a specific gut-to-brain connecting nerve. The model I used to do this was called the TRAP2 mouse. The TRAP2 mouse is a genetically modified mouse model that is able to trap neurons in specific regions of interest. We had two kinds of TRAP2 mice for this experiment. We had a control mouse, and we also had a CCK trap mouse, which was a mouse that was infused with the gut peptide CCK. The two regions of interest that we looked at were the nodus ganglion, which is a small bundle of neurons on the gut-to-brain connecting nerve, and the nucleus tractus solitarius, or the NTS, which is a region of the brain. My job this summer was to slice these sections, stain them with CCK-activated neurons, and image them using microscopes. In the end, we found that there was no significant difference in the amount of CCK-activated neurons in the CCK trap mice versus the control mice. This may be largely due in part to the fact that we had a limited number of time to slice, stain, and image the sections. Nonetheless, this information can help researchers form comprehensive understandings of how CCK is able to activate neurons in the gut-to-brain connecting nerve to further understand the way that they can regulate food intake, glucose levels, and weight gain in individuals that have been affected by obesity and diabetes. Thank you.
Hello everybody, my name is McKean Schaefer, and this summer I worked in Dr. Collins' material synthesis lab. Now, in our modern world, we depend upon electrical devices in almost every facet of our lives. Now, these electrical devices often depend on magnets, but most magnets are based on rare earth metals, and in recent years, we've seen supply chain issues associated with these rare earth metals, and we expect these issues to continue or even increase. My project involves synthesizing magnets without the use of rare earth metals and rather with the use of cheaper transition metals. Specifically, we're investigating making C14 polycrystals, a compound structure known for dense element packing, which we believe can contribute to magnetic properties. We synthesized our compounds using a vacuum arc melting system, and then we analyzed our product using an X-ray diffraction analysis, which confirms that the correct compound was synthesized. Then, we looked to measure the Curie temperature of our magnets using a vibrating sample magnetometer. The Curie temperature is the temperature above which a magnet no longer exhibits ferromagnetic properties. Ultimately, we discovered ferromagnetic properties in 5 C14 compounds, all of which exhibited Curie temperatures above room temperature, characterizing high temperature application like in rockets or in high output computing. Thank you.
Hello, I'm Faiza Shah, and this summer I worked in the Tafali lab studying microbes and researching metabolomics. Metabolomics is the study of metabolites and basically the overall view of what microbes are using as energy sources, what pathways they're taking to produce what in consequence. I was especially interested in the characterization of extremophilic microbes as they thrive in harsh environments. I focused on two of these environments that are namely still unexplored. The first being Martian regolith soil stimulant samples that mimic the Mars environment as there's zero oxygen and high concentrations of perchlorate, a toxic bleach compound, making it extremely hard for plants to grow. We don't know if any microbes are on Mars. We do know there are microbes here on Earth found in sludge water, a similar environment with perchlorates that have adapted to survive. So we ran metabolite extraction tests using analytical tools, FTICR, LCMS, and NMR to gauge a biological understanding of the components found in these microbes and especially understand if perchlorate production is possible, as well as if these microbes produce any nutrients that could make growing agriculture on Mars possible. The second environment I focused on was snake venom. As the microbiota is still very much unexplored, we used the Mojave green rattlesnake found here in Arizona to extract venom and run DNA extraction tests. The problem is there is no kit designed for snake venom, so we use multiple various DNA extraction kits designed for soil. By extracting genomic DNA out of snake venom, we can understand what microbes are there, what they do, how they function, and possibly create better antivenoms. The overall picture of my two projects was the characterization of extremophilic microbes, but the applications apply to astrobiology and medicine. Thank you.
Hello, my name is Brian Stevenson, and this summer I worked in Dr. Seiler's Optical Sciences Lab, which uses lasers to control and study the properties of two-dimensional quantum materials, or in other words, atomically thin crystalline solids. My project focused on assembling and characterizing these two-dimensional materials, whose lattice structures can be rotated and stacked to form a moire pattern, as shown. You can imagine each 2D material as a different Lego brick and put into perspective the many possible varieties of structures to be created. Stacking these materials can be outlined in a 3-step process. The first step is exfoliation, or the process of using Scotch tape to peel flakes of these materials to obtain thin layers. The second step is fabrication, which uses a PDMS stamp, as shown, to pick up and stack these two-dimensional materials. The third step is to optically characterize the complete stack using lasers to verify its quality. I started my project with graphene, a single layer of graphite. I used this common 2D material to refine the fabrication process by optimizing the stamping speed and the stage temperature. After these preliminary transfers, I exfoliated WSE2 and MOSE2 monolayer samples, which are two-dimensional semiconductors, as well as boron nitride, an atomically flat insulator. This culminated in the fabrication of 2 semiconductors encapsulated by boron nitride. I characterized this stack using a technique called photoluminescence spectroscopy, which measures the light emitted by a sample when excited by a laser. As shown in the graphs to the left, the wavelengths and intensities indicate that the samples are high-quality monolayers and that the heterobilayer region between these samples is of high quality and has interesting properties to study. In the future, I can study and characterize the stacking angle between these two-dimensional materials and understand how this affects their optical and electrical properties. Overall, the phenomena that emerge in the twist angles of two-dimensional materials have tremendous potential for next-generation electronic technologies, such as optoelectronics or light interactive technology, and nanoelectronics, or very small devices. Thank you.
Hi, I'm Clarissa, and this summer I worked in Dr. Ledford's Asthma and Airway Research Lab. My lab primarily focuses on the protein called CC16 produced in the distal airways. This protein is anti-inflammatory, antimicrobial, and prevents airway remodeling and immune cell recruitment. Low levels of this protein are also associated with diseases like asthma and COPD. So my research primarily focused on asthma. First, I looked at asthma exacerbations and the differences between asthmatic and non-asthmatic mice. I looked for the expression of CC16 host defense factors as well as the different types of cells in these mice. So what we found was that asthmatic mice had much higher levels of granulocytes than non-asthmatic mice, and we found that our data trended towards asthmatic mice having lower levels of CC16 and host defense factors than non-asthmatic mice. Next, I looked at a potential therapeutic for asthma called C836. This therapeutic was based on CC16 and was administered by lung to asthmatic mice who couldn't produce the protein on their own.
So with this, I looked for immune cells, I looked for histology, including mucus and inflammation, and I also looked for the expression of asthma-related genes. So I honestly found no significant difference in any of the methods of analysis, save for airway hyperresponsiveness using a ventilator. So we found that the mice with the compound had lower airway hyperresponsiveness than non-asthmatic mice, but that was it. And so we think that it had limited success because the drug was administered to the lung instead of to the bloodstream, because CC16 in the bloodstream prevents immune cells from crossing the barrier into the lung, but CC16 in the lung also has protective factors but can't do that. So next we would probably go and administer the drug to the bloodstream instead and look at the differences. Thank you.
Hi, my name is Tanisha Sanil, and this summer I worked in the Ellis lab where we explored the role of holiday junction recognition protein, also known as HJURP. We specifically looked at DNA damage, which is important as damaged DNA can lead to mutations in cancer-causing genes. We specifically looked at ribosomal DNA due to its unusual sequence. Failure to maintain the integrity of rDNA can lead to many human genetic disorders. When the cell wants to perform DNA damage repair, it can do so through a pathway known as the homologous recombination pathway. In G1, growth phase, and HJURP may potentially be involved. In my experiment, I grew cells from a cancer cell line and then transfected them with a gene known to induce DNA damage. As seen by the image on the right, the green fluorescence indicates that the transfection was successful. I then stained for various antibodies and checked for HJURP presence and DNA damage. When DNA damage is induced, nucleolar caps form, bright spots in the nucleoli. In my results, although I did not see clear nucleolar caps, I did see indication of DNA damage, but the HJURP staining was not optimal. In further work, we hope to perform further antibody staining and immunofluorescence. At this point, we cannot conclude whether or not HJURP is involved in G1 activation of the homologous recombination pathway. Understanding HJURP's role in the homologous recombination pathway and rDNA repair can lead to potential insights in the downstream effects of rDNA damage in human genetic syndromes. And by introducing damage into rDNA, we may be able to kill cancer cells, and by studying this process, provide insight into many new cancer therapies. Thank you.
Hi, my name is Sonia Swarup, and I worked in the GEMCorps lab this summer with breast cancer cells. Specifically, we were trying to delete a gene from these cells using a gene editing method called CRISPR. CRISPR works by using an enzyme complex called Cas9, which goes into the cell's nucleus and can cut out and delete unwanted DNA sequences. So there are multiple different ways that Cas9 complex can be delivered into our cell, and our research project looked at which of two different methods was most effective at gene editing. So our first method was called lipopectamine transfection, and it used positively charged lipids to bind to the negatively charged Cas9 complex and bring it into the cell via endocytosis. And our second method was called electroporation, and it used electrical pulses to shock the cell and open pores in the membrane which the Cas9 complex could then enter through. Additionally, we also inserted a drug-resistant plasmid with the GFP gene inside the cell, which is important because we had an electroporation submethod where we added a selection drug to kill off all the cells without the drug-resistant plasmid. And then we could visualize the cells under a special microscope because they glowed green, and then we could count these cells and use this to estimate the number of cells with the actual gene deletion that we wanted. And we found out that 1.6% of the transfection cells and 1.8% of the electroporation cells had the GFP gene, which didn't show a major difference between these two methods, and 31.6% of the electroporation with selection had the GFP gene, which although shows that more cells probably had the gene deletion, they were much less living cells because a lot of them got killed due to the drug selection. And then we ran these cells on a gel, and the double band showed us that the gene editing was successful. So all the electroporation with selection samples had double bands, and then about 60% of the electroporation and transfection samples had double bands. And we can use these results for future projects for better Cas9 delivery into cells and to have more edited cells. Thank you.
Hi, my name is CJ Terrell, and this summer I worked in Dr. Hunter's lab studying the relationship between the leaf-footed bug and the bacterium Cabironia. Many insects have very positive relationships with microbes known as symbiotic relationships. These relationships are often vital to the insect's survival, so the microbes are typically passed down directly from mother to offspring in a process called vertical transmission. This ensures that all generations have access to the microbes. The system I studied is a unique exception to this general trend, as the leaf-footed bug must find and ingest Cabironia from its environment every single generation, or else it faces shorter lifespans and developmental abnormalities. The question we asked was what happens when you delay the insect's access to Cabironia past that ideal developmental stage where they would typically acquire it in the wild. Specifically, how does this delayed acquisition impact the insect's gut in the M4 section, which contains specialized structures called crypts, which house the bacteria. Insects were raised and exposed to Cabironia at varying intervals after the ideal developmental stage, and this Cabironia was labeled with GFP, or green fluorescent protein, so that it could later be visualized. Once the insects reached adulthood, their guts were isolated and imaged with fluorescence microscopy to confirm colonization with Cabironia. After analysis, we found that as the interval of delay increased, the size of the guts decreased, specifically in the length. This indicates that the timing of Cabironia acquisition could be a limiting factor for leaf-footed bug populations in the wild. Additionally, this relationship of the leaf-footed bug and the bacterium Cabironia could be used as a model relationship for more general interactions between animal guts and symbiotic microbes. Thank you.
My name is Lucy Boydell, and I spent this summer working in Dr. Matson's lab with Dr. Logan. As the saguaro decomposes, it becomes a home to many different organisms, one of which is Drosophila nigrosporacula, which is a species of fruit fly. However, the saguaro is also home to Macrocheles sebaceus, which is a parasitic mite that feeds off the blood of these fruit flies. We know that this species of fruit flies will avoid the mites, but we don't know how, so my project was focused on creating a behavioral assay, or test, to determine whether the flies avoid the mites through smell. To do this, I compared two different types of behavioral assays, a choice and a no-choice assay, using a similar species of fruit fly more readily accessible in the lab, Drosophila mohavensis. A choice assay is when flies are given the option between laying eggs on a plate with mite smell and a plate without mite smell. While a no-choice assay is when vials of flies are compared. Some vials contain the smell of the mites and some don't have that smell, and then we compare how many eggs are laid in each vial. I also looked at whether dyed potato media was feasible for egg laying within Drosophila nigrosporacula, as that species can be quite picky on where it lays its eggs. My results showed that a no-choice assay was preferable, as many choice assay trials resulted in no eggs being laid. This may be due to the smell from the mite plates spreading throughout the container. Interestingly, there was a trend of avoidance within Drosophila mohavensis, which had not previously been recorded in this species. However, the results were not statistically significant. The colored potato media was also a success, as it both elicited an egg-laying response within Drosophila nigrosporacula, and those eggs were visible to researchers for the purposes of counting. Now, future research can use this to create a no-choice dyed potato media behavioral assay to determine whether Drosophila nigrosporacula, and then hopefully other species, detect and avoid these parasitic mites through smell. Thank you.
Hi, my name is Elena Wyndham, and this summer I worked in Dr. Andrews' lab, where the main focus was to widen the general understanding of autism spectrum disorder and who it affects. My project focused specifically on calculating prevalence rates and ratios to determine if there's a difference in prevalence rates of autism spectrum disorder among various racial and ethnic groups. I collected my data by using a dataset from the Autism and Developmental Disabilities Monitoring Network, also known as the ADAM Network, from a study put out in 2016. This study included 12,808 children of 4 and 8 years old of various racial and ethnic groups, such as our reference group, which is non-Hispanic white children, Hispanic children, non-Hispanic Asian or Pacific Islander children, and non-Hispanic American Indian or Alaska Native children, among others. Using this data, I created a graph that shows the prevalence rates per 1,000 children split between age with all the racial and ethnic groups that we looked at. In this graph, it is shown that the 8-year-olds have an overall higher prevalence rate than 4-year-olds, which shows that 8-year-olds have a higher diagnosis rate. Using this graph, I created a table that shows whether the differences from the reference group of non-Hispanic white children were statistically significant. In the 4-year-olds, we found that non-Hispanic Asian or Pacific Islander children and Hispanic children had a 1.5 times or higher diagnosis rate than non-Hispanic white children. And in 8-year-olds, American Indian or Alaskan Native children, non-Hispanic, had a 1.6 times higher diagnosis rate of autism spectrum disorder than non-Hispanic white children. Although there were other racial groups that seemed to have a statistically significant prevalence difference, their p-value was above 0.05, which proved that they were not statistically significant. From my results, I proved that my hypothesis was correct and that there is a difference in prevalence rates of autism spectrum disorder across various racial and ethnic groups. My project can direct future research to explore the root cause as to why there are certain racial and ethnic groups that have a higher prevalence difference than others do. Thank you.
Hi, my name is Darren, and this summer I worked in Dr. Pyrus' lab working on the impacts of Alzheimer's on blood vessel function. To do this, we looked at the smooth muscle cells that surround the blood vessel and control its constriction and dilation. More specifically, we looked at the BK channel, which is the large conductance calcium-activated potassium channel on the cell membrane of the smooth muscle cells. The BK channels are both calcium and voltage activated, and I focused on the calcium activation of the BK channel. We did this through looking at the TRPML1 ion channel that's on the lysosome, which is high in calcium concentration. When TRPML1, the ion channel on the lysosome, opens up, we're hoping to see calcium sparks that will then activate the BK channel to allow for blood vessel dilation. We did this by cutting the blood vessel longitudinally and then laying it flat so that we can see the smooth muscle cells. We then dyed it with a calcium indicator to see it fluoresce when there is a calcium spark. We then added a TRPML1 activator known as MK683 in hopes of seeing more calcium sparks. We found that there was no significance in the difference between the calcium spark frequency. However, we did see an increase in the spatial spread, or the size of the calcium sparks, as well as the amplitude or the intensity of these sparks. This can be used in future Alzheimer's research in targeting the TRPML ion channel in hopes of returning normal blood vessel function. Thank you.
Hello everyone, my name is Abiram Chervu and I worked in Dr. Ryan Goodenkamp's lab under the mentorship of Dr. Emmanuel Fonseca. Cichlids are a family of fish known for their rapid diversification, with some species forming in just thousands of years. This makes them ideal for studying evolution and the formation of new species. My research focused on inferring various evolutionary factors that influence two Midas cichlid species, Amphilophus amarillo and A. sagete. More specifically, we focused on understanding their demographic history and distribution of fitness effects, or DFE. The demographic history refers to events that shape a population's evolutionary trajectory, like past population sizes, migration events, and the time when two populations become distinct species. The DFE explores how mutations impact the species' survival and reproduction. Our general method for inferring the demographic history and DFE involved leveraging the fact that the current genomic data of a species is impacted by past evolutionary events, allowing it to be used for inference. We used the DADDY software to infer likely scenarios from the species' genetic data based on different models and optimize those results to better reflect the original data. The results of the models that best reflected the current genomic data of the species were considered the most likely inferences for their demographic history and DFE. Finally, we assessed inference robustness by analyzing fluctuations in the inferences from similar data. Supporting previous research, our findings indicate that these cichlids became distinct species very recently, about 2,668 years ago, in the presence of ongoing migration. Moreover, we inferred very similar DFEs between the species, suggesting that selection impacts them nearly identically. Some limitations of this study include the simplicity of the models used, as well as the potential for different demographic histories and DFEs to produce similar results. Enhanced models and increased genomic information can therefore increase inference reliability in future studies. Overall, we aim to advance our knowledge of cichlid evolution by providing more detailed information about these species and help guide their conservation strategies through a better understanding of their genetics. Thank you.
Hello, my name is Chanmay Dewangan, and this summer I worked on a project under Dr. Nirav Merchant in the Cyvers lab aimed at changing how research scientists manage and share their research datasets. Federal agencies like the NSF, NASA, and the NIH each have their own unique data sharing requirements, which makes it difficult to manage research datasets. Furthermore, data is stored across a variety of locations throughout the internet, which also makes it incredibly difficult to access and manage these datasets because they cannot be moved as easily from one place to another. Therefore, the goal of my project was to develop a new method for research scientists to share and access research data. I used datasets from the Drive Arizona project to test my methods because the project requires large amounts of traffic data to be stored and accessed amongst its team members. Firstly, I created a Python script to obtain metadata from the current platform and then replicated those datasets to a platform called CCAN, which is designed for efficient data management and storing. CCAN also allows datasets to be indexed by Google and also allows for the customization of datasets according to the researcher's wishes. It was extremely important to replicate these datasets into CCAN because the previous platform was not as accessible to the general public. The next part of my project was converting datasets into something called the croissant and DCAT metadata formats. These metadata formats standardize the datasets and overall make it easier for researchers to use those datasets in CCAN. Another aspect of my research was converting CSV, or comma-separated value files, into something called Parquet files, which require less storage while maintaining accuracy at the same time. The last part of my project was creating a user interface so that researchers can easily manage their datasets in CCAN. The user interface serves as the results of my project because it combines all my Python scripts and functionality into one accessible platform. Thank you.
Hello, my name is Akshita and I'm working in Dr. Leroy's computational lab. This summer, I researched the population of adults who have undergone neurotrauma and abuse, specifically traumatic and non-traumatic brain injury and concussion. Traumatic brain injury is caused by an outside force or external impact to the head. On the other hand, a non-traumatic brain injury is caused by internal factors of the brain. A concussion is a mild form of traumatic brain injury which results in a temporary loss of brain function. Abuse of the elderly has become a grave societal issue. Previous research detected elderly abuse among orthopedic surgery patients and found that 0.01% of admissions were due to domestic abuse of the elderly. I used the All of Us database, which contains electronic health records intended to reach 1 million Americans, to analyze the demographics most affected by neurotrauma and abuse. I also looked for signs of elder abuse or abuse of adults at or over the age of 65. I created 3 cohorts for the 3 types of brain injuries, and I had 2 conditions for each cohort. 1, that participants have undergone the injury, and 2, that participants have a history of abuse. My results show that women were significantly more likely to undergo neurotrauma and abuse than men. I also found that out of all races, African Americans were the most likely, closely followed by whites, and Asians were the least likely to undergo neurotrauma and abuse. Although the elderly population was a smaller portion of the data, elder abuse is still existent. I found that out of all affected adults, more than 11% were elderly. Specifically, for traumatic brain injury, over 10%, for non-traumatic brain injury, over 21%, and for concussion, over 6% of affected adults are elderly. This research is crucial as it sheds light on abuse existent in our society and those who are most vulnerable to it. Thank you.
Hey everyone, I'm Shivang Parmar, and this summer I worked in Dr. Lee's lab to create a model that can detect and mask out weeds. Weeds are a big problem in agriculture. Not only do they compete with other crops for resources, but they also harbor diseases and destroy natural habitats. It's estimated $32 billion are lost every year because of them. To make my model, I first annotated a lot of images, by which I mean I drew a rectangle around lettuce and weeds and then labeled it. Then all the annotations were converted into JSON format for an easier access. After that, we wrote a code that would learn from all our annotated images and train a model. After we had an initial model, we tweaked the learning metrics of the model to see if a better model can be yielded. Once we thought that we had the best model possible, we used the model on some test images, and the predictions from those images were then fed to a secondary code which generated masks. The model was pretty good at generating masks, as you can see in these images. The model performed very well on the AP, or average precision test, which is a metric used to see how good an object detection model is. It got a 78 out of 100 on average, and an 86 when detecting lettuce, and a 78 out of 100 again when detecting weeds. This is pretty good knowing our initial sample size was about 300. However, it sometimes did detect false negatives, but this can be improved by adding more images. As you can see, the masks are pretty good, but it struggled when the object was too small. However, overall the model performed very well with a 97 to 99 confidence when it was predicting on some images. This model can be improved further by annotating more images. This model will not only help farmers lower their production cost, but as the production cost will decrease, so will the retail price of lettuce. Additionally, if we can pinpoint where weeds are, we can use herbicides more effectively, which can stop some of the environmental harm that weeds cause. Thank you so much.
Hi, my name is Devin Patel, and this summer I worked under Dr. Tyson Swetnam at Cyvers. I created a reproducible AI-based chatbot with research and educational applications. In recent years, AI, specifically AI chatbots, have become increasingly popular. However, these chatbots often are not trained on current data and hallucinate when responding to a user. I've created a chatbot based on ChatGPT's 4o model that can also be trained on a custom set of files specific to its target use case. Implementing this chatbot follows a simple 3-step process, the first of which involves setting up the chatbot through OpenAI's API and choosing a list of files to train it on. The second step involves setting up a virtual machine to handle messages between your website and the chatbot. Lastly, you need to deploy the chatbot onto your target website. Looking into the financials, I discovered that the average cost per message sent and received by my chatbot is 9.5 cents. This is caused by the length of messages, the number of messages sent, and the size of the files it's trained on. Although some may seem that this price is expensive, it can easily be lowered by limitations on the length of messages and interactions and the amount of messages in each interaction. I've implemented this chatbot onto multiple of Cyvers's online learning workshops where it is trained on the course materials. This is designed to help students in the course ask questions and receive thoughtful answers with resources provided. I've also implemented this chatbot onto my website where it's trained on the resources and files required to deploy the chatbot so it could aid users in deploying this chatbot. Thank you.
Good afternoon. My name is Mavik Padar, and this summer, under the mentorship of Dr. Gandhi Leroy and the Eller College of Management, I was given the opportunity to analyze American susceptibility to traumatic brain injuries, or TBI, using the All of Us database. As traumatic brain injuries continue to grow within American society day by day, it's starting to be more crucial that we understand how and who actually receives these types of injuries. Using the All of Us database and its public health records that are donated by thousands of Americans, firstly, we created a data subset or a data cohort consisting of 8,000 people which have been flagged for traumatic brain injuries by All of Us. Then, using Python and SQL, we were able to then create specific filters in order to analyze my data even further into 2 distinct categories: accidental and purposeful injuries. After analyzing my approximately 7,000 person data group, I was able to see that the main demographic that was consistent in my accidental TBI group consisted of white female seniors who may or may not have gotten traumatic brain injuries through falls or other types of accidental injuries, while on the other hand, those in my approximately 1,000 person purposeful group consisted of white or black males aged from 45 to 64 who have faced militaristic violence, normal violence, or abuse, which have led to the TBI itself. Overall, my results hope to help in actually helping these at-risk populations by raising awareness and also reducing the prevalence of TBI within American populations such as the ones that I analyzed. Thank you.
Hello, my name is Vinay Ramasamy. For my KEYS internship, I worked under the guidance of Dr. Melville Wolgamut in the University of Arizona Bat Lab. My research focused on how bats change their rate of vocalization in an open environment compared to a cluttered environment. Vocalizations are high-pitched sounds that the bat produces and releases into the environment to precisely navigate to its prey. My goal was to find patterns for when the bat was vocalizing in an open room with nothing but its prey, and for when it was vocalizing in a cluttered room with its prey along with other obstacles in the air. To do this, my lab had conducted some trials with the big brown bat species and recorded the vocalizations using a microphone that was placed in the lab. After this, the lab had sent me the large audio files for me to start my part of the project. My first step was to make sure that all of these recordings were set into the MATLAB data processing software. After this, I made sure to go into each recording trial and make sure that all vocalizations are represented in the dataset. Sometimes, since the vocalizations can be too low in volume, they are not represented due to the fact that they cannot pass the threshold of the program. I manually add these in and the data files are ready to go for data processing. Data processing is when I take a lab-developed code fragment and put in my individual files to count the number of average sonar sound groups per trial. Sonar sound groups are a specific pattern of vocalizations that the bat produces right before its hunting sequence. This helps us tell how the bat is about to change its rate of vocalization when it is getting ready to hunt its prey. Overall, as a result, we found that when the bat is vocalizing in a cluttered environment, there are more sonar sound groups due to the fact that it needs a precise navigation to its prey. Our next steps as a lab are to focus on the hunting aspects of each of the trials for more accurate data that we can use to come to a conclusion. Thank you.
Hi, my name is Emmanuel, and this summer I worked in Dr. LaFleur's lab. We researched whether sleep has an impact on cognitive function in older adults, adjusting for cardiovascular disease. This research is important because it can help us understand what factors lead to cognitive decline and mitigate cognitive decline in public healthcare. To start this research, we compiled 3 survey datasets: PAN, ADNI, and the NACC dataset. I specifically focused on the NACC dataset. We then created 3 dichotomous variables: sleep, cardiovascular disease, and cognitive impairments. Sleep and cardiovascular disease were each crafted from a series of subvariables, while cognitive impairment was crafted from scores obtained from cognitive assessments. Using these scores, each individual in the dataset was classified into one of 2 groups: unimpaired and cognitively impaired. The issue was that 2 of the datasets used only one type of cognitive assessment though, and the NACC dataset used a mixture of 2 different kinds of cognitive assessments. Therefore, to establish consistency across our 3 datasets, we standardized the scores of the 2 cognitive assessments in the NACC dataset. After having created the 3 variables, we proceeded with a statistical analysis using logistic regression. Our primary hypothesis tested whether sleep impacts cognitive impairment, as is represented by Model 1, which did not show a statistically significant relationship. However, we did find a statistically significant relationship between cardiovascular disease and cognitive impairment in Model 2, which persisted even when additional demographic variables were added in Model 3, as is illustrated in this prediction plot. As you can see in this prediction plot, as education increases, the probability of cognitive impairment decreases, and as age increases, the probability of cognitive impairment increases. And most importantly, we can see that people who have cardiovascular disease have a slightly higher probability of having cognitive impairment than those that do not have cardiovascular disease. Thank you.
Hello, my name is Amukta Tirupati, and this summer I have been working under Dr. LaFleur's lab, which aims to study the natural process of aging in terms of cognitive decline and how it is accelerated by sleep problems and cardiovascular disease, or CVD. We hope that with our study we are able to provide better comprehensive health care and improve the quality of life in the elderly. To do this, I have taken the Precision Aging Network dataset, or PAN dataset, which is a series of self-surveys and test scores from cognitive function tests. With this large dataset of 473 patients, I have filtered it down to 231 patients by creating 3 yes or no variables, which consists of sleep problems, which is sleep apnea and sleep insomnia, cardiovascular disease as heart disease, stroke, hypertension, and cognitive function as unimpaired or having a mild cognitive impairment, or MCI. With this filter data, we then ran various statistical tests to see whether these 3 variables have a relationship. These tests determined that there is a large proportion of MCI patients with sleep problems and a larger proportion of these patients with the history of cardiovascular disease. However, this difference was not statistically significant. We then ran tests with demographic variables such as age, education, and race group, where the race group was statistically significant, specifically that the Hispanic race group was more likely to have an MCI compared to the non-Hispanic white race group. We hope that with our findings, we are able to mitigate cognitive decline in the elderly in the near future. Thank you.
Hello, my name is Sayan Gupta from Dr. Shabley's lab in the physics department. There's still a lot for us to learn about nanomaterials, which play a huge role in electronic components. This summer, what me and my mentor looked at was the Hall effect in 1 atom thick graphene. The Hall effect is where you take a magnet and place it near a 2D material, which deflects the flow of electrons through that material and changes its resistance to current. What we wanted to see is if this magnet could be replaced with these spiral-shaped molecules placed on the surface of the material. So in order to test this, first we had to create our graphene piece to test, and we did this by peeling it off of graphite with Scotch tape and placing it onto silicon chips. We then choose a graphene piece that we wanted to use, and then in order to attach wires to it, because the graphene is too small, we added contact pads that we could attach wires to and flow current through the graphene. We created these contact pads by placing a film onto the silicon chip with the graphene on it, etching out the pattern we wanted for the pad using an electron beam from an electron microscope, filling that in with gold, and then removing the film, which leaves just gold in the shape of the contact pad that we wanted. Then finally, all we had to do was attach wires, run some tests to compare with previous literature, and measure the resistance as a function of the magnetic field. We didn't get to test the spiral-shaped molecules, but in the future, we'd hope to see that these spiral-shaped molecules act as an extra magnetic field in our graphs. If successful, this would have the potential to create practical superconductors at room temperature, which would help mitigate energy loss in wires.
Hi, my name is Erin Ortiz, and this summer I worked in Dr. Gerba's environmental microbiology lab and Dr. Achilles' water treatment lab on 2 separate projects. In Dr. Gerba's lab, I looked into salicylic acid use as an ingredient in sanitizers rather than current active ingredients which harm the environment. And to do this, we propagated virus, conducted an initial plaque assay to see how much virus there was, treated the virus with different concentrations of salicylic acid for 10 minutes, and then conducted a final plaque assay to see how much virus was inactivated. And what we found was that salicylic acid was effective at inactivating virus. We also found different concentrations to be used in future synergistic experiments, which will test how salicylic acid's effectiveness can be increased when working with other antimicrobials. So for MS2 virus, the optimal concentration would be 0.1% salicylic acid, and for Phi-X174 virus, the optimal concentration would be in between 0.2 and 0.3% salicylic acid for these experiments. And in Dr. Achilles' lab, I looked into the use of granulated activated carbon, or GAC, to remove chlorine from secondary municipal wastewater. And it's important that this chlorine is removed because it can react with organic contaminants to form disinfectant byproducts, which are carcinogens. And GAC is very good at removing this because it has a net-like structure, which makes it great at catching contaminants. So, to test this, we dosed wastewater with chlorine, and then we added both GAC, which has carbon reaction sites to remove contaminants, and catalytic GAC, which has carbon and nitrogen sites to remove contaminants, to the wastewater, and then we measured chlorine removal every 10 minutes until 99% of it was removed. And then we calculated the rate of chlorine removal. And what we found was that catalytic GAC was 26% more effective than GAC. However, catalytic GAC and GAC had very similar performance after the first dose of chlorine. So for future wastewater treatment, people might consider using catalytic GAC over GAC for chlorine removal, just because it is slightly more effective. Thank you.